Hey, Sway.
Hi, Aaron. How are you?
Good. Good to see you.
Good to see you.
How are you doing?
Good. So should I run my slide? Make sure everything is okay.
Yeah, Sway.
Hi, Aaron.
Yeah, test out your slides, please.
Okay, 1 second.
Can you share them?
Yes, sorry. Sure. And does it work?
I can't hear you.
Hello? Can you hear me?
Can you hear me?
Yes, I can hear you.
Okay. Maybe is there something with your microphone settings?
Oh, let me double check.
I heard you before.
Can you hear me now?
Go to Zoom, preferences, audio, and then see your speaker. There you go. Yeah, that's good.
Can you hear me now? Okay.
Yeah, perfect.
Maybe can I share my slide again, just in case?
Yeah, please.
Maybe I'm sharing and then
Yeah, let's try.
can you hear me?
Yes, yeah. And then
Hello?
do the slideshow mode. Yeah.
Works?
Yeah, looks good. Great.
Okay. Stop sharing.
Great.
Okay.
I'm in Shanghai right now.
You went back. I remember
Yeah.
I saw you a few weeks ago, right? Or
Yeah.
3 weeks ago?
Yeah.
Okay. How was the weather? Is it cold?
It's cold. Yeah, colder than California.
Okay.
Hi, Mark.
Hello. Hey, Mark, I know it's a East Coast time. If you want to go first, it's okay for me because it's 5 p.m. for us. For you, it's late. We cannot hear you.
How's that?
Good.
Yeah, I'm happy to go first or second. Either way is fine.
Either way is fine.
Yeah, I think Sway is up first today.
Okay. Either way.
Yeah. Hey, Mark. Can you first? Thanks for coming. And could you test out your slides?
Yeah. How's that? Are you guys seeing the slides or the presenter view?
Presenter mode. So you just have to, yeah.
How's that?
Still
presenter?
Yeah, go use slideshow. Wait, or swap displays maybe. Wait, yeah, that's good.
That's good?
Yeah.
Okay, cool. That should, I just have one video. Let me. Hey, all right. Oh, this thing's going slow. Alrighty, should be in business.
Cool. Yeah, thanks for coming.
Yeah, thanks for inviting me.
How's everything going in Rutgers?
Good. It's been, an interesting couple of years starting up a lab in the middle of a pandemic.
Yeah, it must have been tough. You just started in like 2020?
21.
Oh, okay. Yeah.
Wow.
Yeah, so it's been just about a year and a half now.
Wow, time flies.
It feels like I just started.
Yeah. Sway, how long have you been at Stanford?
5 years.
Mm.
Long
Wow.
time.
Wow. Cool.
Yeah, it has been a while. But I made my presentation short. So make sure I can finish within 25 minutes or 20 minutes.
Okay, cool. Yeah, lots of time for discussion.
So I see that the series is ending soon.
Yeah, coming to an end.
Cool. It's been fun.
Yeah, thanks
You
for
know,
the support.
it's gonna be, there will be a celebration or something, or a conference later in the future, like in person thing.
Yeah, all sorts of things are under consideration. Yeah, definitely we should have some sort of celebration.
Okay.
But it's been going on for about almost 3 years.
Yeah.
Hey, it's long.
Hello. Let's see. Mark, nice to see you. Hi.
When are you coming to China, Sway?
Asking me?
Yeah.
I'm waiting for the summer, maybe summer. And
Okay,
also like
maybe.
my, yeah, summer may be better for kids, right? My son got like so very sick a few weeks ago, and he was in ICU for for a
Nice.
week. I was
Oh
like,
my God.
like chaotic for us.
Yikes. How's he doing now?
He's recovered, yeah.
Okay, good.
Yeah.
I'm looking forward to the ARS meeting, Shanghai.
Oh, yeah.
Finally.
Yeah, it'll be good. I got, so it's a little bit tricky because I got a sponsor last year to pay for the speaker dinner in advance. And now I called the restaurant. I said, hey, remember I paid for this dinner last year before COVID? And they said, well, we don't really know, remember that. And then I said, well, I talked to this person. They said, that person doesn't work here anymore. So I'm just trying to track all that down.
You have the receipt? No?
I have a receipt with like a red stamp and everything. It should be fine, but yeah.
They definitely should receive to tell us everything. So
Yeah.
I know how the finally the quest story goes.
Yeah, should be fine. And then yeah, it'll be really, really good. It'll be good to see everyone.
Yeah, people still start to traveling around within China. So that's fine next month.
Yeah, I'm trying to go to Beijing as soon as possible. Hey, Jen.
Yeah, we're gonna have our final talks
Yeah.
in 2 weeks.
So that'll be the day before Aaron come back?
The day before, I'm coming back on the 25th.
Oh, wow. Okay.
And we have our meeting on the 26th or 27th.
Yeah. All right.
Like moving now, like traveling the US to China every month.
Something like that. It's easy now.
But yeah, definitely. In 10 years ago, usually when we traveling like once a month, back and forth.
Mhm.
Well, we'll look forward to the season's finale. 3 years.
Yeah, we'll plan something good. But then I think there'll be other well, we're still trying to figure out what to do next, but I'm sure we'll do something good.
Mhm.
We're planning it might be sponsored by Neurozoom. I'm not sure yet, but we're planning an in-person huge ALS focused meeting in Shanghai in middle of March. So that should be good.
Expecting mostly audience are, that's pretty short time. Anyone to make it over.
Yeah, it'll be just within China. Well, anyone who wants to come can come, but it'll probably just be within China.
It was delayed for a whole year actually.
Yeah.
Anyway.
But we reserved a really nice auditorium in Shanghai with red leather seats, and we have Juning Yuan giving the keynote. We have the best pizza in Shanghai delivered for lunch. All sorts of cool things.
Looking forward. So where is it? Best pizza?
I can take you there. It's Nanchang Lu Pizza. It's on Nanchang Lu. It's called Joe's Pizza. It's the same Joe's Pizza from New York, but they opened one in China. Yeah.
Got it. Nanchang. Okay.
Do you know where it's right across from the IAPM mall?
Yeah.
I usually go there once or twice a week. It takes about 45-minute subway ride, but it's good. I'm going there tomorrow, actually. So long, if you want to meet there.
Yeah, I try to make it my day. So not my busy as Songjiang, so it's kind of 1 hour drive away.
Yeah, this is a 1 hour drive worth pizza.
Yeah, definitely sounds good.
One of my friends from Hangzhou is coming to town, so I'm meeting them for pizza there.
Nice. Should we get started, Aaron?
Yes, good. Okay. Hi, everyone. Welcome to another edition of Neurozoom. It's good to see everyone. Sort of in the final countdown stage of Neurozoom. And before we get started with the talks today, let me introduce our talks next week. We have Danchang Liu from ION, and we have Jinyoung Lee from Stanford University. And those will be great talks. Please tune in. And then as we're winding down our Neurozoom, our grand finale will be the following week, where me and Zalong will give research talks and then maybe have some sort of celebration about how we were all together over these past 3 years, every week, sharing science and meeting new colleagues and expanding our horizons. So it's been a lot of fun. Okay, so now let's get started with the talks today. And it's a pleasure for me to introduce our first speaker, Dr. Sui Wang. Sui is an assistant professor in the Department of Ophthalmology. She's my colleague at Stanford University. Sui did her undergraduate degree here in Shanghai at Fudan University. Then she went to her PhD at Vanderbilt University, where she started studying gene expression regulation, which controls cell fate determination and differentiation in the pancreas. She had a number of nice papers. She then did a postdoctoral fellowship at Harvard with Dr. Connie Sepko. And there she built on the experience she developed during her PhD in dissecting gene regulatory networks, but now shifted the focus to the retina. She discovered a number of things, among which she had a very nice paper in developmental cell, where she discovered how a transcription factor called BLIMP1 can regulate a binary fate decision between in the retina between rod photoreceptor cell fate and bipolar interneuron cell fate. She pegged this all to a specific cis regulatory module in the BLIMP1 gene, and she showed using in vitro or in utero electroporation and CRISPR in vivo in embryos that if she deletes the specific cis regulatory element, it abolishes BLIP1 expression. She then worked out the upstream regulatory factors that converge on this element. In her own lab at Stanford, she continues to use these types of techniques to study cis regulation. She also builds on some of her experience working in the pancreas from her PhD to sort of study how diabetes and problems in the endocrine system can converge on the retina. So she goes back and forth between this. She develops tools, and she also uses those tools not only to help all of us with our research, but also to help her define gene expression regulation. So really looking forward, Sway, to hearing what you're up to in the latest. Go for it.
Thanks a lot, Aaron. I will share my slide. Is this a presenter mode?
Yeah, you just want to switch. Once you play, and then you can switch.
This?
Perfect. Yep.
Work. Perfect. Thank you, Aaron and Zilong and all the other organizers for giving me this opportunity to share our research with you. As Aaron has introduced, I was trained as a developmental biologist, so I was not trained in neuroscience. But we have some common questions between the pancreas and the CNS, so I would like to address and study. So my lab has two major topics we're very interested in understanding and address. We are interested in learning how different cell fates or neuronal cell fates are specified in the central nervous system, and also how different types of neurons or glial cells respond to insults in adults or when there's challenges to the system. We address these questions from a specific angle. As Aaron has introduced, my expertise and my training is on transcriptional regulation. So we would like to, also here is a very simple diagram of a cell. So when there's inputs, it can be developmental cue or disease insult. It will induce a cascade of interacting proteins and other signaling events. And often there will be transcription factors activated, which goes to the nuclei, bind to the cis regulatory modules or elements in the genome and activate transcription, which then you get new proteins and other events to cope with the challenge. So we're specifically focused on two elements in the system. One is the cis regulatory modules, where the transcription factor binds in the genome on the DNA, and also transcription factors. So they interact with each other. I would like to mention this is not a simple like one-way direction. There are a lot of feedbacks between the interactions. So we call this a gene regulatory network with two key components we focus on. One is transcription factor. The other one is the cis regulatory modules in the genome. And we specifically address these big questions, using a model system, which is a vertebrate neural retina. For those of you who may not familiar with the retina, here is a very simple cartoon schematic. So in the retina, we have photoreceptor cells, which are light-sensing neurons, rod and cones. And we have interneurons, horizontal cell, bipolar cell, amacrine cell, which relay the signal to retinal ganglion cells, which then send the signal to the brain. I would like to mention, we think the retina is not only a camera. It probably works as a computer. It not only captures the light and send the information to the brain, it also do feature extraction and then do calculations for our vision signals. So it's very important organ or a tissue for us. So we're interested in this system, retina, is not only because there are a lot of eye diseases or retina-related disease we would like to conquer or solve, but also for us, the neuroretina is a great model system to address a lot of questions. So here I'll give you an example of the major techniques or methods we use in the lab. So it's really easy to perform genetic manipulations in the mouse retina in vivo or in rodent retina in vivo. So it's a little hard for the brain, but for retina it's very easy. So we can basically stick a needle loaded with plasmid. In this case, is a CAG GFP. CAG is a ubiquitous promoter, drive GFP expression. We can stick the needle into a live pup, newborn pups, and stick the needle to the subretinal space between the retina and the RPE, which is a supporting tissue. And we just apply electricity, just zap the animal. And then what we get is we can have the DNA, the plasmid DNA, basically mini preps, delivered into the retina cells, and it will express whatever you have in your plasmid. So here shows like two very nice patch we got after electroporation. And then when we section the retina, you can see a lot of cells can be labeled. So this way, using this technology, we're able to do a lot of genetic manipulation to understand genetics or gene regulation. And so specifically, my lab is interested in two major directions. One is we would like to understand how different types of retinal cell fates are determined during development. So we know that the multipotent retinal progenitor cells will exit the cell cycle, become newly born post-mitotic cells. At this stage, their cell fates are still plastic. They're not determined. We can still switch their fate. And later, they will be determined to be specific cell types. So we're very interested to understanding what's going on during this process. Additionally, we are interested in understanding how different retinal cell types respond to disease insult, acute injury or chronic injury, such as diabetes, which I studied when I was a graduate student. So today, for the sake of time, I will only focus on the first, the retinal development or retinal cell fate determination direction. And just show you some of our published data and unpublished data. And hopefully in the future, if I have a chance, we can talk about the retinal responses to disease. But our logic is the same. So we do a similar experiment, which is use different models, development or disease situation. Okay, so how different types of retinal cells, their fates are specified during development. This has been heavily studied in the past 3 or 4 decades. So we know a lot, actually. So here are the major retinal cell types during development. So the x-axis is a timeline in mice. So embryonic stage and postnatal stage. Here's a newborn. We know that different retinal cell types were born at a specific time point during development in a stereotypic order. You always have ganglion cell, horizontal, and cone cells born at the embryonic stage, and later followed by rod, bipolar cell, Mueller glia cell. They were born relatively late during development, and the retinal fate specification finish or differentiation finish by the first week of birth. So this order is very stereotypic, and we also know it's mainly transcription factors that regulate this fate decision. This is a very stereotypic fate decision. So here I list the transcription factors, which are known, discovered in the literature, and we already know that play key roles for the specification of each of those cell fates. I list them here. And if you look at some of them, you can see there's transcription factors which play roles in different places during development. It's not only one specific cell type. It's not like one cohort of transcription factor control one fate. They're like jumping all over the place. So for example, OTX2 control rod, cone, and horizontal, and also bipolar specification at different time points during the development. So currently we know there are a lot of transcription factors regulate the fate decision during development, but we really don't know how they wire up in this network to control this competence change during development and make sure they are going to produce ganglion cells at early time point and produce bipolar or Mueller glia cell at late stage. So that's the big question we would like to know. So again, just to emphasize the question we're interested to address is how is the expression of this different transcription factor regulated, and how do they wire up and form the network to precisely control the fate decision during development at specific time points. So today, I will give you a specific example of focusing on the regulation of transcription factor OTX2, which is a homeobox transcription factor. I'll give you an example. Hopefully, we can induce or excite some discussion on this big questions. So OTX2 is a famous transcription factor for retinal development. We know it regulates multiple aspects of retinal development. It is expressed in retinal progenitor cells. Here, just a staining shows that early post neonatal stage is expressed in the progenitor cells. In mature retina, OTX2 has low expression level in photoreceptors, high expression level in bipolar cells. So you can see the expression pattern become restricted. And then there are loss and gain of function analysis of OTX2 has already been done and published. So we know that during development, if we delete OTX2 gene, completely delete, we lose photoreceptors, bipolars, and we have more amacrine cell. It's a fake switch. So more interneuron. Amacrine cell is an interneuron. Interneuron, the number will be go up, and photoreceptor and bipolar cell will be gone. However, if we reduce the level of OTX2 gene, we have more photoreceptors, less bipolar cells. If we have high dose of OTX2, we have, for example, you overexpress OTX2, you have less photoreceptors, more bipolar cells. So it's a very interesting transcription factor. It's required for the specification of both raw photoreceptor and bipolars, but the dosage controls the cell fate. So with this in mind, we go forward to ask, how is the OTX2 expression regulated in this cell type specific and stage specific manner in the retina, right? So it's expressing multiple cell types at different stage, at different cell types. So we employ a very easy way. So to understand gene regulation, we think, okay, because the transcription factor bind to the cis regulatory modules, what if we identify the cis regulatory modules first, and then figure out what are the transcription factor bind to it? Then we know how OTX2 is regulated. And right now, there are a lot of technologies to let us predict cis regulatory modules, such as ATAC-seq, cut and run or cut and tag, and also other method like DNA-seq, like a more old method. So we can predict some of the cis regulatory modules, and by analyzing those cis regulatory modules, we can figure out, hopefully we can figure out the transcription factor which regulate this cis regulatory module. So that's what we did. So based on the ENCODE project, the published data, we identified all the DNA's hypersensitive region, which is the open region in the genome, which are highly possible to be cis regulatory modules. So here we show at different stage of retina development, and the peaks correspond to the region which are open in the genome, which has a higher possibility to be cis regulatory modules. So we get many of those peaks, and then we did a simple experiment. We PCR out all the different peaks, 25 of them, and clone them into a reporter plasmid, which has the minimum TATA promoter drive the expression of GFP, iris AP. AP is alkaline phosphatase. It has give you this strong color as a reporter gene. So without any insertion of cis regulatory modules, the TATA cannot drive any expression in the retina. But if you insert a candidate CRM or candidate cis regulatory module, which has enhancer activity, it will drive the expression of GFP or AP. So we electroporate all the candidate reporter plasmid into the retina together with the electroporation efficiency control plasmid. So here's our results. It's basically black and white. So in the control situation, so the cherry indicate our electroporation worked in the retina. So each of this is a retina. In the empty control, you don't see any AP staining. But some of them, 5, 7, enhancer 5, 7, and 9 and 20 and 15 gave very strong signal. Means that those are active cis regulatory modules that can drive strong expression. So we start to study those guys. This is the X-plan experiment, which is kind of very fast. So we verify the findings in vivo and focused on 05, 07, and 09 enhancers or cis regulatory modules because they show very robust and interesting pattern in vivo. So what we did is we electroporate the reporter plasmid, carry different types of CRMs or cis regulatory modules with GFP, and co-electroporation CAC-BFP electroporation efficiency control into a newborn. And then we wait for 3 days or 14 days and we harvest the retina and see what is the expression pattern. So this time we're using GFP as the reporter for the enhancer activity. So there's a lot of data here. I will just summarize it. So at P3, all 3 enhancers are on in retinal progenitor cells. But at P14, their expression patterns start to diverge. So O5 is highly expressed in bipolar cells and a little bit in photoreceptor cells. O7 only in photoreceptor cells, not in any other cell type. And O9 is off. So O9 was on in progenitor cells, but it was off in adult. So here's just a quick summary of what we see. So O5, O7, O8, which set the 3' of OTX2 gene. They regulate the expression or probably regulate expression of OTX2. And then they have specific activities in different cell type. O5 is more active in bipolar cells. O7 is only active in photoreceptor cells, and O9 active only in retinal progenitor cells. So then we want to know what are the transcription factors bind to those different enhancers and regulate the OTX3 expression in different cell types at different stage. To do this, we also have a very simple pipeline. So we chop or bash the enhancer or CRM into different fragments, and then we incorporate them into our report, each of the pieces into our reporter plasmid and do electroporation, zap them into the retina and see whether it can drive AP expression. It's also black and white results. So O5-8, this little piece can drive very strong expression. Others cannot. So we narrow it down to around 100 base pair lengths. And we learned that we always have, around 100 base pair is the minimum. If we chop O5-8 into a smaller piece, it doesn't work. So 100 base pair, around 100 base pair is the minimum. Then we do predict the transcription factor binding site based on the sequence, and we mutate each of the potential transcription factor binding site and see which one can abolish the activity. So here we find one which can, when we mutate the OTX2 binding sites on the sequence, the activity is completely gone. So that's how we figure out the thing. And then we do other EMSA and the chip and other experiment to confirm the binding of this OTX2 transcription factor with this enhancer. So I just skipped the study or the results we did for 07, 09. Just to summarize, we found that OTX2 bind to O5, CRX bind to O7, and SOX2 bind to O9. And those 3 transcription factors are not foreigners for us. We already know those 3 play essential roles for retina development. We already know they have been studied, especially for O5, it's self-regulated. So we also incorporated our data with cell type specific ataxic data published in the literature. So here just a summary of our results. So in progenitor cells, O5, 7, and 9, all 3 elements are open, and transcription factors OTX2, CRX, and SOX2 are all present or expressed in the progenitor cells. So they all together regulate the expression of OTX2. In raw photoreceptor cells, the expression level of CRX is high. OTX2 is very low. SOX2 is not expressed in photoreceptor cells. So CRX is the driving force which drives the expression of OTX2. But in bipolar cells, the situation is different. O7 is not open by ataxic. O7 is closed. So even though bipolar express a little bit of CRX protein or transcription factor, it's actually using the O5 OTX2 combination to drive the expression. In Mueller glia cells, which is interesting, O5 and 7 are closed by ataxic. They are not open in the genome. Even though SOX2 is expressing Mueller glia cells, but somehow is not using the O9 open enhancement to drive OTX2 expression in Mueller glia cells. This is something we haven't figured out why. The transcription factor is there, the enhancer is open, but OTX2 gene is not open or not expressed in Mueller glia cells. There may be additional mechanism contribute to this regulation. So I show you the retina. So here I summarize all the reported data on the regulation of OTX2, including the work published in the brain. So the The colored ones are the previously identified enhancer of OTX2 at different brain tissue or during development. And the black ones are retina. And the black ones with the green box is the ones enhancer that are active at embryonic stage. So you can see none of them, except for here, which is a promoter region, almost none of the distal enhancer or distal cis regulatory element actually overlap with each other. Nobody overlaps. So all the retina and brain developmental enhancers are segregated. They're using different regions. So we found this is very interesting, how this landscape was set during development. So they use different pieces of DNA or different regions to regulate the same gene in different tissues. So just a quick summary. So we think the cell type and stage-specific gene expression in the retina or in neuronal tissues is controlled by 3 factors. One is availability of the upstream transcription factors. Second is the accessibility of the cis regulatory modules within your genome. Your genome has to be open at a certain region to be regulated. And also the dosage of transcription factor is also very important to determine the outcome of gene expression and then the fate decision. So we think the system has some advantages, as because we know that we have a very limited transcription factor pool in our genome, about 1,500 transcription factors, but they control so many different events during development. And most of the transcription factors are redeployment at different tissue. So most of the transcription factors that control pancreatic beta cell differentiation or pancreatic other cell types differentiation in the pancreas are also control cell fate decision in the retina. That's why I kind of switched field. I felt like, oh, it's kind of the same sets of transcription factors are doing the fate decision, but the outcome is very different. So right now we are focusing on 2 questions. One is how the cell type or stage-specific chromatin accessibility is established during the development. How this dynamic of the chromatin openness or openness of the chromatin is established during development. We felt like that's very intriguing and interesting. We're trying our best to chase after this question. And another question we're interested in addressing is, how still we have a specific example OTX2, but can we put other transcription factor on the map and understand how they systematically regulate the fate decision during development? And at the end, my last slide, I would just to see that. So today I kind of focus on the 6 regulatory elements and transcription factors regulating fate decision and transcription factors. We are also doing similar type of study to understand how different retinal cell types respond to diabetes. So what are the cis regulatory elements that respond to this disease insult, such as diabetes, and regulate gene expression and contribute to disease development? In addition, we also using those cell type specific cis regulatory elements or cis regulatory modules, and we put them into AAVs and generate, hopefully generate AAVs that can target specific cell type in the retina and in the brain. And this is some of the work I've already published. If anyone's interested, we are a lab with expertise generating different types of virus. With this, I would like to thank the people who did the work. Candice did the retina development work, and Chenghui did the diabetes work. I didn't have a chance to mention. They all left the lab to become graduate student or faculty member. Here's a current labs. And then we would like to thank our collaborators, Patrice Ford, Tom Gunner from University of Michigan, Stanley Qi, Junning, and Lu Chen from Stanford. And we're funded by NEI and ADA. Thank you.
Thanks, Wei. Great talk. And we're open for questions. I think Kai-Wen has a question.
Yes, please. I'm curious.
Hi, I leave the questions in the chat room. So I'm just curious. I'm always curious actually. So transcription factor plays a very important role in determining which genes can be turned on. But what is there anything known to regulate as an upstream signals to regulate the presence of the of this?
Transcription factor. Yes, that's a great question. So we have layer 1 and layer 2 and layer 3 go up, and the initial events we are not sure, but we think maybe the initial, in the hierarchy, the initial event could be openness of some of the genomic regions, make them accessible to transcription factors. For example, but we don't know who are those things. Some people use the word pioneer factors. So those factors, transcription factor, can open the genome at certain regions, maybe in a sequence-specific fashion. But in the retina development, we're not sure. But I'm sure everyone knows about the IP, stem cells, Yamanaka factors. I think those are probably some of the factors as a higher at the hierarchy to open the genome, and then it's a transcriptional cascade, layer 1, layer 2, and go. So it's a great question. We don't know, and we're from the bottom up, so it's kind of hard to figure out the entire network. That's the goal.
I'm not familiar with the gene regulation, the domain. So I'm always curious. Those IPS, those transcription factors that can determine the cell fate. So they have to be translated, right? They have to be transcribed and translated. So there must be some environmental factors, or just what kind of cues to turn on this? We do not think it's environmental factors, or environmental factors play a very limited role. The reason is we think this transcription factor cascade is very intrinsic, at least for retina development, because if we put early retinal progenitor cells in a late environment, it still gave rise to the early cell fate. If you put a late retinal progenitor in a young or embryonic environment, it still gave rise to the late cell fate. So they're intrinsically wired. We think the environmental challenge or environmental changes may kind of bias the ratio a little bit, but doesn't change the fate. That's something we think is very cool, how this transcription cascade or clock is set. So it's so scientific, and it's almost the same for every device, for everyone. Thank you.
So I have a couple of questions. So in other gene regulatory network situations, you'll have the same transcription factors regulating different choices and there'll be binding sites for those factors that have different affinities. So the concentration can matter. I'm wondering, in your situation, you presented that you have different transcription factors in the different cell types and different binding sites to your modules, but do you see ones where there's the differentiation is based on the affinity for the binding site of the same factor?
We don't have direct evidence, but we suspect there are. For some, for maybe O5, there's some affinity issue there, but we don't have an easy assay to test, especially in vivo situation, to test the affinity of transcription factor binding with in cancers, right? Because you deliver a plasmid to the cells, we're missing this in vivo affinity. So that's a great question. We would love to have something or a method so we can measure the affinity of transcription factor and CRM binding affinity. But I agree with you, and it's similar to other situations. The affinity definitely play a role, but we haven't have a easy way or smart way to tease it out.
Got it. Okay. There are a couple more questions. If you want to unmute, you can ask them.
I think I can see them. Do I have time, or I can type the answers in the chat box? I do not want to delay the.
Yeah, maybe that's a good idea, Sway. Maybe you can reply to everyone to answer these questions in the chat, and then so long.
I'm just curious to know how you're planning to trace the effect of perturbation of any transcription factor or the regulatory modules in the adult phase or any other injury condition in the later stage.
In later stage?
Yeah.
So based on sorry, I don't have time to show the data. So what we do is, in the late stage, we're not looking for fate changes. We're more looking for, what are the cis regulatory modules which respond to disease. For example, become open when there's a disease challenge like diabetes, and what are the genes they regulate which contribute to the development of retinal disease. So we're now looking for fate decisions for the late stage. Do I understand answer your question?
Okay, thank you.
Yeah.
Right. Great. Thanks so much, Sui. And Zilong, do you want to introduce Mark? Looks like Zilong might be frozen.
That's all right. I can introduce myself.
Sure. Let's see what's going on with him. Okay, yeah. So I normally spend long hours preparing these introductions and reading all papers and doing research and finding things. I didn't do that for you because Zilong was going to do it. So I apologize. Maybe he's going to try to log back on.
It's no problem.
Zilong's back.
Oh, is he back?
Yeah. Hi, Zilong. Are you? Zilong, we're having trouble hearing you. All right. Sorry, Mark. Maybe could you just introduce yourself?
Absolutely, not a problem. No worries. Let me get this. Are you seeing the slides now?
Yes, looks great.
Okay. So thanks for the invite. So I'm Mark Rossi from Rutgers University in New Jersey. I'm an assistant professor in the Department of Psychiatry located at the Child Health Institute in New Jersey. And so I did my undergrad work in Michigan, and then I moved to Duke for graduate school, working with Henry Yin, studying basal ganglia circuits and motivated behavior. And then I did a postdoc with Garrett Stuber at University of North Carolina, and then the University of Washington, where I looked at hypothalamic circuits and their relationship with feeding and obesity. And I started my own lab in 2021. And yeah, we work on feeding and obesity stuff. One of the things that I'm really interested in and that my lab studies a lot is the relationship between brain circuits and energy balance. So we know that energy balance is largely controlled by the brain through interactions, bidirectional interactions with the periphery. And so this is just a super oversimplified schematic of that. But essentially you have signals from the periphery, such as from the GI tract and the adipose tissue, coming into the brain, acting through periventricular organs to sort of influence the neural activity and to shape behavioral output. And so the part of the brain that we think a lot about in my lab is the lateral hypothalamic area. So the lateral hypothalamic area is a part of the brain that has for a very long time been thought to influence feeding behavior. So all the way back in the 1940s and 1950s, researchers started performing lesions in this area in and around the hypothalamus. And they noticed that when they lesioned selectively this part of the lateral hypothalamus, the dark shaded regions here in this example, lesions to that area profoundly impaired feeding behavior. So basically all voluntary food-directed behavior ceased with these lesions. And this has been replicated across species for many decades. And they also noticed that if you do electrical or chemical stimulation of the very same areas, you can induce feeding in well-fed animals. And so over the subsequent 60 or so years, a lot of people spent a lot of time working on this problem, trying to understand the relationship between the lateral hypothalamus and motivated behavior. And so what they came up with is this idea that the lateral hypothalamus is sort of very well situated to integrate various multimodal signals and integrate those signals and send filtered output to diverse downstream targets. So this is just a schematic representation of some of the inputs coming from basically all over the brain, cortex, hindbrain, you name it, going into the LH, along with direct input from circulating signals. And similarly, the neurons in the lateral hypothalamus send output to a variety of different areas, including many of the same areas that it receives input from. So it's really well positioned to have a big impact on behavior. And so now, going on probably around 15 years ago or so, people started using the modern sort of viral and genetic tools to disentangle what the different cell types are within the lateral hypothalamus. And what we're seeing is that there are a lot of different cells there. So primarily there are two basic neuron types. You have your glutamatergic and GABAergic neurons. And these comprise about greater than 80% of the neurons located there. And so they seem to have rather opposing functions. So work from Garrett Stuber's lab showed that these GABA and glutamate populations within the lateral hypothalamus have opposing influences on feeding and food intake and body weight regulation. So they found that if you selectively stimulate the glutamatergic, so the VGLUT2 expressing neurons in the lateral hypothalamus using channel rhodopsin, so this is a light-gated ion channel. Basically, if you turn these cells on selectively in hungry mice, they will pretty much immediately stop eating. And when you turn the light off, they go back to eating, and you can sort of do this for a while back and forth. In a sort of opposing manipulation, they used caspase to genetically and selectively ablate these LH VGLUT cells. And what they saw is that the mice with the caspase ablation showed a propensity for weight gain when given access to calorie-dense chow. And the GABAergic manipulations resulted in essentially the exact opposite phenotype. So stimulation of these cells caused robust feeding, and ablation of them caused resistance to high-fat diet-induced obesity. So this suggests that these neurons influence both acute feeding as well as body weight regulation. And so that's where I came in at the start of my postdoc. I was kind of interested in first understanding just what types of cells are in the lateral hypothalamus and if they're affected by something like diet-induced obesity. So to do this, we did high throughput single cell sequencing from the lateral hypothalamus in control and diet-induced obesity mice. So they were maintained on one of these two diets for about 10 weeks. And then we took LH tissue punches and ran them through a single cell sequencing pipeline. And we ended up with about 20,000 cells, roughly 50-50 from each of these two groups. And what we found is sort of all of the canonical cell types you would expect from brain tissue, so neurons and glial cells. And we saw that we got large populations of these GABAergic cells and glutamatergic cells, making up the majority of the neurons. And we did not see any de novo addition or subtraction of cell types in the high-fat diet group. So it seems like all the same types of cells are present in both groups. But we did find that there were lots of genes that were altered in the high-fat diet group. So this is the signal-to-noise ratio, and each of these dots represents a different gene. And so these are all the different clusters. And you can see there are lots of genes that are altered in the high-fat diet group across all the different cell types. And so when we filtered those genes looking for highly expressed genes that were also had a large magnitude of change, we saw that a lot of those other clusters sort of dropped out, and we were left with the GABA and the glutamate clusters showed the greatest number of genes after this filtering. And so when we looked at the types of genes that were altered in these groups, what we saw is that the GABA and the glutamate cells were the genes that were enriched for each of these were genes that were enriched in the VGlut cells were de-enriched in the VGAT cells. So essentially, it suggests that there's opposite changes going on in the VGAT and the VGlut clusters. And we saw that many of these genes were associated with things like synaptic activity and ion homeostasis. And so that suggested that there may be functional remodeling happening within these circuits in response to this high-fat diet-induced obesity. And so to further get at that, we used in vivo 2-photon calcium imaging in the lateral hypothalamus. So we focused first on the glutamatergic population. And so we injected the genetically encoded calcium indicator GCAMP into the lateral hypothalamus and then implanted a microendoscopic Grin lens over top of the LH. And the mice are then positioned, so they're head fixed and they're stuck under the 2-photon microscope. And for these experiments, there's not really much of a task. The mice are just sort of hanging out in a tube, and there's a sucrose delivery spout directly in front of them, and we randomly deliver drops of sucrose and look at the activity as the animals are voluntarily consuming that sucrose. And so this is an example of one of these videos, and you can see there's flashes of activity that are associated with the sucrose consumption here. And so the first thing that we were interested in is understanding what the response to sucrose consumption or food consumption in general is in these cells and whether or not it varies, but with satiety state. And so to do this, we just, again, gave the mice drops of sucrose, and we recorded them on consecutive days, either when they're ad lib fed or when they were overnight fasted. And this is an example of one of these cells. What we saw is that there were a lot of cells that showed a greater magnitude of response to sucrose consumption when the animals were fed versus when they're fasted. So this is an example of one of those cells here. So each of these rows now is a trial. And when we look at the whole population, so now this is about, I think about 550 cells, what we see is that it's a bit more of a mixed bag. So certainly it's not an overwhelming, uniform trend, but we saw that there were a lot of cells that showed this same pattern where they were more active to sucrose consumption when the animals were fed. And on the population level, that shows up as this reduction in the magnitude of the response. And it also shows up as a change in the proportion of neurons that are significantly responding to this task. So we have a reduction in the number of excited neurons and an increase in the proportion of inhibited and unresponsive neurons in the fasted condition. So together, this suggests to us that these cells are consistent with their optogenetics results previously. These cells are negatively regulating feeding, and when the motivation for food is very high, the activity of these cells tends to be dampened. So then we took advantage of one of the great aspects of this in vivo 2-photon imaging approach, which is the ability to longitudinally track cells for very long periods of time. So this is an example of an experiment where we tracked the activity of individual neurons for 12 weeks throughout the onset of diet-induced obesity. So we took the mice and we split them into control diet or high-fat diet groups, and the high-fat diet group unsurprisingly gains significantly more weight. And we can reliably resolve these cells for the entire duration of this experiment. And what we saw is that in the control group, the cells start out being responsive to sucrose consumption. So again, the sucrose consumption is shown here in this dashed line. So these red signals indicate increased activity that's happening after the animal starts a bout of sucrose consumption. And we see that these cells maintain their responsivity across this experiment. So the same cell is in the same position in all three graphs. But when we look at the high-fat diet group, what we see is that there's lots of cells that start out being responsive when the animal's on their chow diet. But very quickly after they go on to the high-fat diet, we see that their activity is greatly suppressed. So they no longer respond to that sucrose consumption. And this is just the the average here. And so what we think is going on is that these cells are under normal conditions acting as a brake on feeding. So they come online after an animal starts a bout of food consumption, and they act to restrain that food intake. And we think that one thing that may be going awry in the brain facilitating overeating is the progressive loss of this brake on feeding. And that may be a contributing factor to things like diet-induced obesity. And so when I started my own lab, one of the questions that I was very interested in is understanding what are the underlying circuit mechanisms that are contributing to these results. And so one obvious candidate to look at is the inputs from the bed nucleus of the stria terminalis. So GABAergic inputs from the BNST to the lateral hypothalamic glutamate cells. The BNST is known to selectively target those cells, and activation of that pathway facilitates food intake. So that's inhibiting the LHV GLUT cells, promotes food intake. And so this was work done by Kuldeep Srivastava in my lab. And so we just wanted to first sort of probe the parameter space of what's going on with this type of effect. And so the first thing that we did was to inject channel rhodopsin into the BNST, and then we're sticking optic fibers over the lateral hypothalamus. So this allows selective activation of this pathway between the BNST and the LH. And so what we saw is that so the task here is, again, we're just taking the mice and we're delivering random drops of sucrose solution, and we're delivering laser light to activate these axon terminals within the LH during times in which the animal is not typically consuming the sucrose. So we're activating the cells in the context of sucrose, but immediately prior to the actual delivery. So during a time in which the animal would not typically be licking very much. And so what we saw is that indeed the activation of this pathway facilitated consumatory behavior and sucrose seeking. So what you're looking at here are graphs of, these are raster plots of the lick data. So each tick mark represents licks, and the reward delivery is happening at the 0 point here. So typically the reward is delivered and the animal performs like a bout of 6 to 10 licks to consume it, and then they usually stop licking. But we're delivering this laser stimulation at either 10, 20, or 40 hertz during this blue shaded region. So it's happening before the reward is actually delivered, during a time where they typically would not be performing many licks. And what we see is that we can reliably induce licking during this period. So you can notice the increase in licking during the shaded region. But what we also saw is that when the animals are fed, they are much less sensitive to this stimulation. So on the bottom row is when the animals have been overnight fasted, and you can see that the same amount of stimulation yields significantly more licking behavior at these higher frequencies. And so that's just shown here. So what we think is happening is that fasting is sensitizing this system to make the animals more sensitive to the stimulation. So we get at these intermediate frequencies, we see this, this strong increase in the rate of consumatory behavior in response to licking that's absent in the YFP control group. So they show no effect of laser stimulation. And so just to sort of verify this in vitro, what we did was to perform patch clamp recordings within this pathway. So we injected channel rhodopsin into the BNST and a dio-M cherry into the lateral hypothalamus in V-GLUT-Cre mice. And so then we are patching onto the V-GLUT cells in the lateral hypothalamus and looking for connectivity. And what we see is that, consistent with previous reports, this pathway is largely GABAergic. So we can block the light-evoked responses by applying gabazine. So it confirms that greater than 80% of the connected cells are receiving monosynaptic GABAergic input from the BNST. And then when we look at only those cells that are identified as receiving monosynaptic GABAergic input, what we see is that the amount of inhibitory input from all sources is greatly increased in the fasted condition. So we see an increase in the rate of spontaneous iPSCs onto these LH V-glut neurons in the fasted condition, and no changes in the rate or amplitudes of the EPSCs. So again, it suggests that there's greater inhibition of, within this pathway in the fasted state. And so the next thing that we did was to look at how this behavior changes with high-fat diet-induced obesity. So to do this, we performed the experiments that I just described, and then we put the mice onto a high-fat diet for 3 weeks, and then we redid the experiments in this sort of on their way to being obese. And then we put the mice back onto chow for 3 weeks, and then we did the experiments a third time. And so, you know, for the sake of time, I'm only showing a snippet of the data here, but what we saw is that in the high-fat diet condition, and this is at a time in which we know that the V-glut cells in the LH are being suppressed, we see that the BNST to LH stimulation is much less effective after 3 weeks of high-fat diet. So you can see here in the baseline chow condition, we're able to evoke a lot of sucrose seeking with laser stimulation. When we put the mice onto the high-fat diet, it's pretty much completely abolished. And when we put them back onto chow, it rebounds to similar levels as the baseline, but the pattern of results doesn't look quite the same. So you notice that the pattern of the licking is actually quite a bit different. And so we're still exploring why that might be right now. But yeah, this is just showing at the population level, we see this reduction in the sucrose seeking in the high-fat diet condition. And so to summarize, this is sort of the working model that we have right now. So under normal conditions within this BNST to LH V-glut pathway, we have when the animal is ad lib fed and not terribly motivated to seek food, we have relatively low levels of this BNST input, which yields greater levels of LH V-glut output. When we fast the animal, we see that sort of flips. We get increases in the BNST inhibitory drive onto these LH V-glut neurons, which suppresses their activity. And we think that under normal conditions, this is sort of operating within, sort of a normal range that it switches back and forth between these various states. In obesity, what we saw is that the LH V-glut cells are significantly downregulated, or their function is downregulated. We still are working on testing what is happening to the BNST VGAT cells in this case. But what we think is going on is that there may be a disconnect where the system that normally switches between these two fed and fasted conditions in sort of a seamless manner, we think there may be some sort of disconnect here where this balance is being dysregulated and maybe facilitating overeating. And so that's sort of where we're approaching this question about the circuit mechanisms from, this is one project, and we're also looking at other circuits related to this as the LH receives lots of diverse inputs. So this is certainly not the only thing that I think is going on, but I think it's one important aspect of it. And the other thing that we're looking at is we're trying to understand the role of circulating factors. So there's a lot of evidence to suggest that lots of peripheral signals can act both directly and indirectly on these LH and associated circuits. And so that's something that we're following up with now and trying to really get a handle on in the lab. And we're also combining these methods, so optogenetics and 2P in vivo 2P imaging to selectively activate and inactivate ensembles of these cells during behavior to understand the specific role for these pathways in behavior. And so with that, I just want to thank all the people that were involved. The first part of the talk was done in Garrett Stuber's lab during my postdoc in collaboration with Marcus Basiri. And the latter work was done primarily by Kuldeep Shrivastava and Vikshar Athreya in my lab. And my thank you to my funding, and I'm happy to take any questions.
It's a beautiful talk. Now it's open for questions. Can everybody hear me now?
Yes.
Thanks. Beautiful talk.
Thank you.
Questions? Hi, Andrew. Andy. Can you unmute yourself? Unmute. Do you have?
Yep, I'm unmuted. Sorry, I stepped away in the middle, so I hope I didn't miss this. Have you looked at different these responses in different times of the circadian cycle, and does it change if you have?
That is a great question. So we haven't systematically studied it. We deliberately try to do all the feeding experiments at like the same point within the light cycle. But yeah, I would not be surprised if there are different effects based on, like, certainly if you were to do it like in the middle of the dark cycle versus in the middle of the light cycle, I would expect to see differences. We're actually, so in my postdoc, we did everything in the dark phase, so during the animal's active phase. In my current lab, because we don't have that luxury of having a reverse light cycle vivarium, we're actually doing these all during the light phase, and we're seeing actually similar effects. So qualitatively similar at least.
Hi, Mark. I have a question, but on the high-fat diet conditions. So what do you think is the circulating factors, blood sugar or some neural input to activate the BNST neuron to inhibit reward seeking?
I think it's probably both. So,
okay.
So I think that there are likely circuit level effects. So the BNST and the LH both receive input from, say, AGRP neurons, as well as other neurons that are known to be, critical mediators of feeding. So I suspect, and those cells also receive input from circulating signals. So there's probably both circuit level effects, so upstream neuronal signaling, but also we and others have shown that there can, at least in certain situations, there can be direct effects of things like leptin and ghrelin and glucose and probably insulin as well on these LH circuits. So that's something that I'm really excited to keep working on, but we don't have a solid understanding of it yet.
Great. Thank you. Okay, now Yu, another feeding expert.
Hi, Mark. Yu Fu from Singapore. Very nice talk. I wonder, you must know the RHA-VGLUT2 neurons project to many different regions. So for the BNST connection, is there any preference?
In terms of the LH cells, where those are going to? Yeah, so we don't know yet. I would say that so far, it seems like they are probably not the habenula projecting cells. So there's a large portion of the LH-VGLUT cells that project to the lateral habenula. We don't have any direct evidence for it, but based on the observations in identified habenula projecting populations and what we're seeing now with the cells, the VGLUT cells that are receiving the BNST input, it seems like it's probably not a large overlap. But beyond that, we don't know. It's something that we're working on.
Okay, and also a general question is like, apparently BNST plays a role in modulating feeding behavior, but what exactly is BNST neurons? What are they sensing, and what is the conceptual role of these guys?
Yeah, I think that's a great question, and that's something, historically people have really looked at the BNST in terms of stress and anxiety and alcohol related things, but it's clear that it has the ability to influence food intake, and that's been shown by a bunch of different groups at this point. I'm not sure. It's unclear to me if it's like whether or not the same cells that are responsible for these effects on the lateral hypothalamus, if these are the same neurons that are receiving input from, say, the AGRP neurons, or if they are, if they're different populations. So one of the things I would love to do is do some transcriptional profiling of that projection and try to understand if there's how they fit in with the other identified BNST populations that have been shown to be involved in other behaviors. So we don't yet know if it's like a separate population.
Great, thanks.
Great, thanks. Now, any more questions? All right. So great. Now, thanks, Mark and Sui, for great talks today.
Thank you.
See you to final last Neurozoom. Great, thanks. Thanks, Mark. Sui, great talk.
Thanks for inviting me.
Good to see you, Mark. Thanks, Sui.
Thank you.
See you
Thank
soon.
you, Aaron and
Excellent.
Zilong.
Okay.
Right.